Hello, everyone. This is Mr. Friedhoff. I'm going to be starting the next chapter with you, which is chapter 13, and it's going to talk about genomics. When we talk about the genomics of biology, we're talking about the complete set of DNA. And you can see our picture here is just looking at all the A's, G's, T's, and C's that somebody has. In this chapter, we have a couple different sections, and the first section is going to kind of relate back to what we learned in chapter 12 when we talked about protein synthesis. So that being said, let's get into 13.1. This is going to be about mutations. Now, we've talked about mutations a little bit before, and mutations are just any change in the sequence of nucleotides in DNA. Now, this can be a single nucleotide or even as large as a chromosome. So the severity or the amount a mutation occurs can really vary. We're going to look in the next section at gene mutations. We're going to see that these gene mutations are going to cause differences in the protein structure. So we talked a lot about how proteins are produced and their function in cells. Now we're going to see what happens when these proteins have mutations in them, and what happens to the how that results or how that affects the cell in general. So remember that mutations are caused by mutagens. These are just physical or chemical agents that can cause a change in the DNA. It's going to be like smoking, UV radiation, X-rays. But we also have spontaneous mutations where whenever we go through replication, sometimes that replication isn't perfect. And what can happen is the DNA can mutate by itself. So this happens not regularly, but it's something we actually kind of want. I know it sounds kind of weird, but mutations aren't a bad thing. Mutations are just changes in DNA. Sometimes they can be bad, sometimes they can be good. So these mutations are left in the resulting DNA from replication, just because sometimes we want mutations. We're going to talk about that in the next couple chapters when we talk about evolution. Now, just like I talked about before, mutations cause a change in the DNA sequence, which can cause abnormal proteins to be produced. So you can see here our normal gene produces a normal protein. However, again, whenever we mutate that gene, sometimes we can get an abnormal protein or no protein at all. It depends on the mutation, depends on the gene, depends on a lot of different things. But basically what I'm trying to get across in this slide is that mutations cause differences in the genome. Or your gene's DNA, whatever you want to call it, which then can cause changes in the protein. And this is actually where we're going to end 13.1. Hello, everyone. This is going to be the second section in chapter 13. This is where we're going to start talking about something called gene mutations. In the last section, we talked about how mutations are just changes in the DNA. Now we're going to talk about certain types of gene mutations. And the first type of gene mutation is going to be called a point mutation. This is where there's a change in a single nucleotide. One nucleotide is changed. That's why it's called a point. Think like a point on, anything. Point just means one single location. What we're going to see is there's a couple different types of point mutations. So there's a couple different ways that these point mutations affect proteins. Because remember, our whole goal here is to look at how DNA is going to make a protein. And when we change the DNA, we're also going to maybe change the protein. And that could have significant just repercussions based on the protein and how it changes it. So let's talk about the different types of point mutations that we see in this section. The first type we're going to see is just a substitution mutation. This is where, again, one nucleotide is just flip-flop. You can see C here used to be a T, so that's just a substitution mutation. This is just where one nucleotide is changed. What happens is this substitution can change in different ways, or this substitution can change the protein in different ways. The three different ways that a substitution mutation can change a protein is the silent, missense, and nonsense. In a silent mutation, when that substitution mutation occurs, there's actually no change in the amino acid. So if you look here, we have serine, valine, proline, tyrosine. If you look, this nucleotide here was changed. However, the amino acid sequence is still the same. That's why it's known as a silent mutation. Because even though we change the DNA, the protein doesn't change. In a missense mutation, this changes the amino acid. So you can see here, we changed the C to an A. That changed proline to threonine. So again, in our original DNA, we go serine, valine, proline, tyrosine. In our mutated one, we went serine, valine, threonine, tyrosine. So that mutation, when it's a missense mutation, when that mutation changes the amino acid, That's called a missense mutation. And our last mutation is a nonsense mutation. This is where the amino acids change to a stop. Again, here's our regular mutation, or this is our regular DNA. It goes serine, valine, proline, tyrosine. Now in a nonsense mutation, you can see it goes serine, valine, proline, stop. This one drastically changes the protein because it's not all formed. Our last kind of group of mutations in this section is going to be known as a frame shift mutation. And this is where it's going to change the reading sequence or the codon sequence of a gene. What happens is in a frame shift mutation, basically that codon sequence is either shifted to the right or it's shifted to the left. And as you can see here, the amino acid sequence is going to be changed for all of the amino acids. This is drastically going to change the amino acid or the protein sequence that we see. Now, what we see in the frame shift mutations is there's only one of two different types. It's either when you add, which is called insertion, or delete, where it's called deletion. So again, when you add one in, that's called an insertion. You can see it just shifts everything over. Or when you take away, that's called a deletion. That's a frame shift mutation because it is, again, changing all of the codons after that where it's either inserted or deleted. I'd like to show you, this is like an analogy where here's the original, I want to say protein sequence, but this is kind of like an analogy with a protein. So the sentence says, the boy cut his lip and ate the hot dog. In a missense mutation, we're changing one of the letters. So it says the boy cut his fip and ate the hot dog. You can see that you kind of still get the gist of the sentence, but it's just a little bit off. In a nonsense mutation, you can see it just ends. It just ends halfway through. The boy cut his, and you don't know what actually happened. So that's a nonsense where a nucleotide or an amino acid is changed to a stop. In a silent mutation, it just is the same. You can see the boy cut his lip and ate the hot. Yeah, it just sounds like the same, the original sequence. In an insertion or deletion, these are again frame shift mutations. You can see it says the boy cut his slip and that. It just makes no sense. Same with the deletion. We took out the D here, so now it reads the boy cut his lip and it just makes no sense. That's why frame shift mutations are so much more detrimental to proteins than any of these. It's because they just totally throw everything off. Hello, everyone. This is going to be chapter 13.3. It's going to be about the Human Genome Project. Just a reminder, remember, chromosomes are made of DNA, and each chromosome has many genes within each of the chromosomes. You can see here, this is a karyotype. If you remember that back from chapter, I think that was chapter like 9 or 10. This is a karyotype. This is the complete set of chromosomes for an individual. These chromosomes are made of DNA. However, sections of those DNA are called genes. A genome is the complete set of genetic material for an organism. So think about all of the DNA in all of the chromosomes for a person. What does that sequence look like? The complete set of DNA is called an organism's genome. And this genome for humans and all eukaryotic organisms lives inside the nucleus. Because remember, chromosomes are in the nucleus, DNA is in the nucleus. That's where our genome consists. And every single cell has a genome in its nucleus. I know that's not going to sound weird. Yes, every single cell has 46 chromosomes in its nucleus. Now, the Human Genome Project is a project or kind of like an organization of companies and governments and just a lot of different organizations that took time back in the 1990s, took over 10 years to sequence the first human genome. Now, again, computers and technology back then wasn't as good as it is now or wasn't as fast as it is now. So this took a long time. It took over 10 years for one person's, just one person's genome to be sequenced. That's learning what the nucleotides are from the first nucleotide to the last. And it costs over $3 billion. $3 billion with a B. Now it takes less than 24 hours and less than $1,000 for us to do this. So it's dramatically come down in price in the past 25 years. But the first person that we did took 10 years and took $3 billion just because we didn't know how to do it at first. Now we know a lot better ways of finding out somebody's genome. You can see what this looks like. So they take the DNA, they make many copies of it, they break them apart, and they sequence those breakages to figure out what that sequence looks like. This is called shotgun whole genome sequencing. It's kind of taking apart these sections of DNA and learning what the DNA nucleotide sequence is. You can also get partial genome sequencing. This is just like a commercialized view of it. But basically, this is just smaller portions of your DNA that we can still kind of learn a little bit about. But I show you in class my 23andMe that I did years ago. There's Ancestry.com and something called Illumina. But these are partial. These aren't the whole genome that you may have. Just a little bit to end here. For humans, we have 3.2 billion base pairs in our genome. For humans, we also have 25,000 genes. And if you look, it sounds like that's a lot, but in humans, again, we had 46 chromosomes. We don't even have as many chromosomes as a dog. Humans have, again, 3.2 billion base pairs. There are some plants that have like over 150 billion base pairs. So it's not that we're the most, it's just that how our species evolved, we have 3.2 billion. And 21,000 genes, about 21 to 25,000, it's actually less than like rice that has 38,000. So it's just not that we are the most, it's just how our species evolved. Hello, everyone. This is going to be the fourth section in chapter 13. This is going to be about gene regulation. When we talk about genes, remember that we're talking about DNA. And something that's interesting to learn about DNA is that only about 2% of the DNA in your body actually makes proteins. You would think that a lot of your DNA makes proteins, but it's actually less than 2%. The other 98% regulates the 2%. When I was in high school, I learned that this DNA, this 98%, was actually called junk DNA because we just didn't know what it did. Through years and years of research, we found out that this 98% actually regulates the 2%. So only a very small percent of our DNA codes for proteins. Now, we have things called transcription factors. These transcription factors are going to be proteins that help either express or stop transcription. So again, a repressor is a transcription factor that will stop transcription because certain cells don't need to be making all the genes. Activators will help transcription occur because we need certain cells to make genes. So this is just one way that your genome is going to be regulated. So either these transcription factors that are just proteins, they either help stop or help activate certain genes in certain cells. All right, the last section here in chapter 13 is going to be about chromosomes. And this is where we're going to talk about, again, DNA in the form of a chromosome. So these are in our cells, specifically in our nucleus. And we learned in other chapters that DNA makes up a chromosome. However, there's also going to be something called a histone. And these histones are basically just proteins that help regulate gene expression. They help also package the DNA into these nicely packaged chromosomes. So these little kind of bluish greenish proteins, these are just histones that the DNA is going to wrap itself around to form these chromosomes. In this section, we're also going to learn about something called telomeres. Telomeres are the ends of chromosomes that are going to be repeats of DNA that don't actually code for DNA. What happens is these repeats are cut off accidentally during replication. So what these repeats do is since they don't code for any DNA or they don't code for any proteins, these DNA can be cut off and not actually damage the vital DNA here in the middle. So what happens is after every replication, some of the ends are cut off, and that's not good for the chromosome. It's not good for the cell, obviously. But what that does, it allows the innermost portion of DNA to be protected. And what we typically see is people with longer telomeres live longer, and people with shorter telomeres don't live as long because without these telomeres, the DNA becomes so damaged that the cell can no longer divide.